Okay, I think we can get started. So if I may, I just quickly introduce Daniel. Good morning, everyone, and thank you all for joining the seminar. It is my great pleasure to introduce our distinguished invited speaker for today's seminar, Professor Daniel Razansky. I know Daniel and his work for many years, and Professor Razansky is a world-renowned expert in biomedical imaging, and currently he holds the chair of biomedical imaging at both the University of Zurich and Zurich in Switzerland. He also serves as director of the Preclinical Imaging Center. Professor Razansky obtained his degrees in biomedical and electrical engineering from Technion Israel Institute of Technology and finished his postdoc training at Harvard Medical School. Before joining the faculty in Zurich, he was a professor of molecular imaging engineering at the Technical University of Munich and Helmholtz Center of Munich in Germany. He is widely recognized for his groundbreaking research in biomedical imaging, specifically in multispectrum optotomography and hybrid optoacoustic ultrasound, which has led to numerous of biomedical imaging technology that has been successfully commercialized and utilized in research labs and clinic facilities around the world. He has won multiple awards from some prestigious organizations such as European Research Council, NIH, as well as the German Innovation Prize. He is also a fellow of IEEE, SPIE, and Optical Society. So we're indeed honored to have Professor Razansky share his insights and expertise with us today. So without further ado, let us welcome Daniel for his seminar.
Thank you, Sean. Thanks everyone for attending my lecture and allocating time to it. I'm very grateful for the invitation and the opportunity to tell you about our recent work on optoacoustic imaging methods for biological discovery and clinical diagnostics. So in a few words, our group is located in Zurich, Switzerland, where we are strategically affiliated with two universities. So the Faculty of Medicine at the University of Zurich, and also the Department of Electrical Engineering at ETH Zurich. And this obviously gives us access to world-class engineering expertise of the ETH, and also excellent opportunities for biological clinical collaborations at the University of Zurich. And we are also very interconnected across the two universities and contribute to, well, now more than 5 competence centers, primarily the Neuroscience Center Zurich, the Skin Integrity Consortium, the ETH AI Center, and another joint Max Planck ETH initiative for learning systems, and also Excite the Zurich Center for Imaging Sciences. So there we extensively collaborate on various technical and algorithmic development projects, and also on the application side of things, for example, in the fields of neuroscience, cardiovascular diagnostics, and skin research. Okay, so when it comes to biomedical imaging in general, it's always associated with those two important metrics, namely the spatial resolution and penetration depth. So the different imaging modalities are usually presented in this kind of simplistic plot. So if you want the microscopic resolution, you will have to give up resolution and also to give up the penetration and also the field of view. But in some cases, it's not as simple as it sounds. So for some imaging modalities, there are also physical barriers that are difficult to overcome. So in the case of optical microscopy, whatever you do with with it, it would be quite impossible to penetrate several centimeters into the tissue non-invasively, just because of photon scattering and absorption. So same holds for other methods like, for example, positron emission tomography. It would be quite challenging to achieve microscopic resolution in deep tissues. So even though it penetrates very deep, and you can do whole body human imaging, but due to some physical limitations of the method, the resolution will be quite poor in this case. For some other imaging modalities like MRI, you can increase the field strength, and then if you scan long enough, maybe a few hours, you can still get down to even microscopic scale resolution and also penetrate quite deep. And same for X-ray. But then you have to give up something else, which is going to be the temporal resolution, because this is going to be quite long. So as I mentioned, so temporal resolution or imaging speed is definitely another critical factor in biomedical imaging. So if you're just looking at very slow stationary things like anatomy or organism development, you don't really care about imaging speed, so you can run your scans for hours. When you're interested in visualizing drug distribution or contrast agent kinetics, you can still live with relatively long scans of minutes or hours. But there are many other dynamic biological processes where imaging speed is critical, for example, flow of cells or heartbeat. So here you need to image dynamics on a sub-second scale. And there are even faster things like neural activity where millisecond scale temporal resolution is required. So depending on your application, it's always about this trade-offs between speed, resolution, and depth. And what is also important is that you would usually choose a different imaging modality to cover different spatial and temporal scales, which is quite problematic because then you also get very different contrasts. So ideally, you would like to do imaging at various scales with the same imaging modality. And this is exactly where optoacoustic imaging steps in with the promise to bridge between all these different spatial and temporal scales. So optoacoustic imaging is a truly hybrid method that synergistically combines light and sound. And hybrid means that we use short pulsed laser beam for the excitation of the tissue. However, what is being detected are those tiny ultrasound vibrations that are generated due to absorption of light by different tissue chromophores. For example, oxy and deoxyhemoglobin. So in this case, the imaging contrast will be optical, or more precisely, it would represent optical absorption, but the spatial resolution can be made either optical or in superficial tissues, or alternatively ultrasonic if you're reconstructed images from deep tissues at, say, centimeter scale penetration where the light is fully diffuse. So we can basically enjoy both from rich optical contrast and also high resolution in deep tissues. So in optoacoustic imaging, the intrinsic contrast stems from absorption by different endogenous chromophores, such as oxy and deoxyhemoglobin, melanin, fat, water, collagen, and others. And basically whatever is dark in the tissue would generate strong signal. So for example, blood. The highest penetration depth is of course achieved in the near infrared spectral region where blood absorption goes down. If you're using several illumination wavelengths, you can then essentially do optical spectroscopy of tissues with optoacoustics, similarly to pulse oximetry. But you can do it also with high resolution in 3D and also in a label-free way, right? Because you don't need to inject anything to sense, for example, blood oxygenation. In terms of instrumentation, optoacoustic imaging can be done in 3 basic modes. In the optical resolution microscopy mode that you see on the left, the light beam is tightly focused into a spot on the tissue surface, and for each pulse of light, a single ultrasound detector is commonly used to detect the generated optoacoustic signals. And then the light beam is scanned across the sample to acquire 2D or 3D images. And obviously, this approach only works for limited depths of up to about 1 millimeter in tissues where the light can be effectively focused. In the second case of the acoustic mesoscopy, so here in the middle, the light beam is no longer focused and the image is formed instead by using an acoustic reconstruction. So in this case, you can image deeper because you don't rely on ballistic photons. So you can usually image up to a couple of millimeters into absorbing and scattering tissues. But the resolution in this case is much worse than in the former case of microscopy because the resolution is acoustic and not optical anymore. So both microscopy and mesoscopy share the common problem of being quite slow because you need to mechanically scan the light beam and in most cases also the ultrasound transducer in order to form images. So the third approach, termed optoacoustic tomography, would use multiple ultrasound detectors arranged on an array to detect the generated optoacoustic signals at multiple locations simultaneously. So in this case, for every light pulse, the images can be formed very fast through the so-called tomographic reconstruction. So basically, in this way, you can do real-time 2D or even 3-dimensional imaging. But for that, you need many detectors and also some complex electronics, which makes the setup much more expensive. So let's see what kind of images can be generated with optoacoustic microscopy first. So in this case, the excitation light beam is focused. So clearly blood is very absorbing, so you can nicely visualize all the vasculature without any extrinsic labeling. So basically label free. So for example, we recently used this approach to track healing wounds in the mouse skin longitudinally, so over a period of up to 2 weeks post-wounding. And here on the right, you can nicely see all the angiogenesis going on down to a capillary level resolution. And what is also nice about this approach is that you can image a field of view of several centimeters, which is not really possible with a regular confocal orthophoton microscope. So obviously, this kind of optical resolution optoacoustic microscopy only works up to a depth of about 1 millimeter, where the light beam can be effectively focused. And again, the method is quite slow because you need to scan the excitation light beam and also the ultrasound detector mechanically along the 2 lateral dimensions to form volumetric images. So as I mentioned, the optical resolution microscopy approach is very limited in terms of depth. So the next thing you can do to enable, for example, deeper imaging into human subjects, so you can do mesoscopy, and for that you would need to use an unfocused light beam, and then you do an acoustic reconstruction to form the images. And here you see examples of images that were acquired from psoriatic patients with optoacoustic mesoscopy, where you can immediately recognize the different vascular morphology in patients versus normal skin. And all this can be done completely non-invasively up to a depth of several millimeters, which is not really possible with other optical microscopy, for example, with 2-photon or optical coherence tomography and so forth. Again, the limitation is the imaging speed because you need still to mechanically scan the light beam and the transducer. So depending on the effective field of view and the number of acquired voxels, it may take somewhere between a couple of minutes or even 20 minutes or half an hour to get those kind of images, especially if you work with several wavelengths to do spectroscopy. So this is obviously not suitable for monitoring fast biological dynamics and can also lead to severe problems with motion artifacts. So in order to solve the imaging speed problem, so around 2009 or 2008, we started developing the so-called multispectral optoacoustic tomography, or MSOT in short. So MSOT uses multi-wavelength illumination of the tissue where each pulse of light comes at a different wavelength. And then images are reconstructed for each wavelength separately using parallel tomographic data acquisition and reconstruction. And at the final step, we perform the so-called spectral unmixing of the image data, which is done pixel by pixel or voxel by voxel. And for this, we use the information on the absorption spectra of the different chromophores present in the tissue. So again, for example, the oxygen, deoxyhemoglobin, melanin, or any other chromophore that or fluorochrome that was a contrast agent that was injected into the tissue. Here you see an example of Alexa fluor dye with a peak absorption at 750 nanometers. So the final result in this case of MSOT is the spatial distribution of individual molecules in the tissue, as shown here on the top right images. So this technology was eventually also commercialized and won the German Innovation Award in 2014. And here you see some examples of the different functional and molecular imaging applications that were performed with the MSOT technology so far. So there are many hundreds of examples actually, so I'm just showing a couple of representative ones. So on the left, you see an example of our recent tumor oxygenation study where we achieved high resolution mapping of tumor hypoxia entirely non-invasively and without using any contrast agent. So with MSOT, you can nicely differentiate the tumor rim from the normoxic and hypoxic areas, and also visualize oxygen gradients across the tumor, and also, for example, look into responses to oxygen stimuli. Another example you see in the middle, it's a study looking into kidney function that was done by a group at the University of Liverpool in the UK. So they injected a contrast agent and imaged the kidney of a mouse over time. So the kidney perfusion dynamics in this case was quite different in healthy kidneys versus nephropathic mice. So this is an example of kidney metabolism study. And last example on the right is a molecular imaging study that was done at the University of Louisville in the US. And this group developed smart liposomes that specifically target pancreatic tumor cells. And in this case, they used MSOT to visualize specific uptake of the agent at the tumor site versus unspecific clearance through the liver and kidneys. Okay, so more recently, so let's say the last 7-8 years, we also invested quite some effort to really boost the imaging performance and develop truly volumetric multispectral optoacoustic tomography technology. So the basic idea is to use this kind of spherical matrix array transducers that can capture the generated optoacoustic responses simultaneously from multiple projections around the object. And in this way, we can achieve fast volumetric imaging of large tissue volumes, such as, for example, the whole mouse brain. And here we use again an unfocused light beam that illuminates the entire imaged volume at once. And for each laser pulse, we simultaneously record a large tomographic data set, which is sufficient for reconstructing an entire 3D volume. So in this way, we have essentially a very fast imaging method that can achieve more than 100 volumes per second imaging rates. And it's basically only dependent on the pulse repetition frequency of the laser. The spatial resolution in this case is limited by ultrasound diffraction and available bandwidth of the transducer. So it would be typically in the 35 to 200 micron range. And the effective penetration mainly depends on the laser wavelength, but usually it would be up to several centimeters in the near-infrared spectrum. And here on the right, you see a picture of the probe with a coupling membrane that we typically use for human imaging in a handheld mode. So one of the first demonstrations of this technology was done in a human breast. And here you see an example of imaging sessions from a healthy volunteer. So basically, the handheld probe is slowly moved along the skin surface, and the volumetric images. So here you see maximal intensity projections along the 3 axes from the breast, and those are rendered in real time, basically at video rate, using parallel implementation of 3D reconstruction on the GPUs. Again, what you mainly see in those images are blood vessels, but also some breast parenchyma and fatty tissues. One limitation is the effective field of view, which is limited to about 1 cubic centimeter for this particular configuration of the spherical array probe. So you would need to scan the probe to acquire some large tissue volumes in humans. So in the follow-up studies, we also applied volumetric EMSOT in mice, particularly looking at several brain disease models, such as epilepsy, stroke, and Alzheimer's. In this particular paper, we were able to track the propagation of epileptic seizures across the entire mouse brain. So basically, the seizures were induced by injecting 4 aminopyridine agents into the cortex, and we then constantly recorded EEG signals in order to recognize when the seizure begins. And with the volumetric EMSOT, we could do high-resolution 3D monitoring of the changes in both the oxygenated and deoxygenated components of the signal. So in this way, it was possible to clearly see the seizure in 3D as it initiates in the cortex, propagates toward the thalamus, and then returns back into the cortex, basically closing the so-called thalamocortical loop. So this is not something you can easily do with, for example, fMRI, because it's not sensitive to oxygenated hemoglobin component, and also pretty slow when you do 3D imaging of the whole brain of the mouse. We also recently used this technology to look at neurodegenerative disease models. So in one recent local collaboration here in Zurich, we were able to study the progression of Alzheimer's disease non-invasively at the whole brain level. And for this, we used amyloid beta targeted probes. So basically, the probe is injected intravenously and specifically accumulates in the plaques, in the Alzheimer's plaques over 24 hours. And we saw an average increase of about 30% in the plaque load over 1 month of disease progression from 14 to 15 months of age. So keep in mind the resolution in this case is quite high, so it's about 100 microns, so not something you can do with fluorescence imaging because the signal gets completely blurred over the 7 to 10 millimeter depth of the mouse brain. So here you see an example of epifluorescence images from the same mouse model. So far, I've shown you that optoacoustics can be used efficiently for imaging brain hemodynamics and accumulation of targeted molecular agents. However, it could also become even more powerful if we manage to make it sensitive directly to, for example, calcium or voltage dynamics in the brain. So in this Nature Biomedical Engineering paper that we published together with a group of Shai Shoam from New York University, we basically exploited the sharp changes in the optical absorption of GCAM6 fluorescent proteins in response to calcium changes in the neurons. And then we looked at the corresponding changes in the optoacoustic signal. So in this way, we managed to record calcium activity non-invasively and also in 3D over a field of view covering the entire cortex of the mouse with 20 millisecond temporal resolution. There is one caveat though, because the one problem remains that GCAM protein absorbs light at 488 nanometers, where the light penetration into mammalian tissues is quite poor because of strong blood absorption. So we couldn't penetrate for more than 1.5 millimeters into the brain. So in comparison, if we could do imaging at 680 nanometers or longer wavelength, optoacoustic can easily cover the entire mouse brain. So to overcome this problem, we are now actively looking into development of near-infrared shifted calcium proteins. And the first such near-infrared shifted genetically encoded calcium indicator, which is termed NearGecko, was reported in this Nature Methods paper, where which we co-authored with the groups of Robert Campbell and Ed Boyden. So in this particular study, we still didn't do it in optoacoustic mode, but only in fluorescence imaging mode, because the response of this particular first generation indicator was not strong enough to be detected with optoacoustic imaging. So hopefully we are now also testing the second and third generation of near-infrared calcium reporters to hopefully see also the optoacoustic responses from the deep brain. So maybe a bit different example of how this imaging technology can be used for diagnostic and therapy monitoring purposes. So many of you are probably aware of this fast evolving field of medical microrobotics or nanorobotics. So basically the big promise of this field is that one day we could have this smart tiny robots moving through the patient vasculature and precisely manipulate it with magnets or acoustic fields. And in this way, we could, for example, deliver drugs very accurately, eliminate tumors, fight infections, do microsurgeries, or maybe even provide some precise diagnostic information non-invasively. So just name it. So very futuristic vision, I would say. But currently, the main problem is that in order to freely pass through the vasculature, those robots must be made very small. So let's say 5 to 10 micron or even less. And currently, there is no way to detect such small particles individually while they circulate deep in the large living organism. So this was exactly the goal of our recent collaboration with a group of Metin City from Max Planck in Stuttgart. And the paper was out last year in Science Advances. So what we did, we basically conjugated micron-sized microrobots with both nickel and gold layers, so they can be magnetically manipulated, and at the same time also detected with high sensitivity using a volumetric optoacoustic imaging system. So here on the right, you see a movie showing real-time imaging of those 5 micron-sized microrobots freely circulating in the mouse brain, which is a quite big leap forward for translating this exciting medical microrobotics field into real life and human applications. So I previously mentioned that spherical array probe are quite limited in terms of the field of view. So the field of view that it can do in real time is about 1 cubic centimeters. So this is just sufficient to image a single organ in real time. For example, the beating heart of a mouse. But nevertheless, you can also quickly scan the probe around the mouse and acquire this kind of beautiful whole body images in a couple of minutes. And we call this technology spiral volumetric optoacoustic tomography, or SWOT in short. I think I'm not able to switch my slides anymore. Do you still hear me?
Yes, I can hear you.
Okay.
The slides is not moving.
Right. Okay. So I think I have to restart the presentation.
Sorry. Here we go. We can see it now.
Okay.
Yeah.
Sorry about that. So I'm back on track, I hope. In the most recent version of this SWOT method, which we called SWOT, which is single sweep volumetric optoacoustic tomography. So we managed to squeeze the whole body scan time down to 1.8 seconds. So according to my knowledge, this would be the fastest preclinical whole body imaging modality. So you can really start looking into not only the biodistribution of contrast agent, but also at how the contrast agent perfused through the entire body in the blood vessels, individual vessels, and so forth, more or less in real time, and at very high spatial resolution. So maybe to say a few words about the importance of, all the inverse theory and reconstruction methodology behind all those images, which is also very important for quantification. So it's probably easier to demonstrate it again with images. So generally, there are, of course, many different reconstruction algorithms that are used in optoacoustic imaging. And what most people use is the so-called approximated back projection formulas. So the problem with those approaches is that the resulting images are not very accurate, and they may even contain some voxel values which are negative, so it doesn't really have a physical meaning. And in this case, if you try to do, for example, spectral unmixing for oxygenation, you end up with most of the brain containing meaningful values or empty values. So we developed an alternative way of reconstructing optoacoustic images, which is called semi-analytical model-based algorithms. And they provide accurate reconstructions which are made without analytical approximations. So as you can see, in this way, you get meaningful images where the blood oxygenation can be accurately quantified across the entire mouse brain. So in this particular paper, for example, we were able to look at hemodynamic changes in orthotopic glioblastoma tumor after administration of vascular disruptive agent into the mouse, and also quantified over time. So development of accurate image reconstruction, acquisition and reconstruction approaches is important for many other reasons. So many people in the field, for example, try to combine optoacoustic and ultrasound imaging in one device. And at the first glance, it seems quite easy. Just take a simple linear array transducer used in commercial B-mode ultrasound scanners, add some illumination fiber to it, and you're ready to go. But the optoacoustic images you get with such an approach are of very poor quality. So here, for example, you see the ultrasound images in gray, superimposed with some colors representing oxygenation in mouse placenta. And it's really hard to recognize any clear anatomical structures in the optoacoustic data, right? In all those colors, it's just some speckle noise. So I very much doubt the oxygenation values are very meaningful in those images. And the main reason for such discrepancy compared to the nice images I was showing previously, it's the poor tomographic coverage of the linear arrays. So here you see that, so you basically have very limited view tomographic geometry with only 48 degrees of coverage available around the focal zone where the object is located. So to better convey the point, look at those cross-sectional images from a mouse acquired with transducer arrays that have different tomographic coverage around the object. From 60 degrees on the left, which is basically the linear array geometry, up to 270 degrees with almost a full ring coverage around the mouse. So you still get quite reasonable pulse echo ultrasound image quality with for all those configurations, even with the limited view linear array. But the optoacoustic image quality is very poor in this case, right? So you can't even clearly distinguish the surface of the mouse anymore. So ideally, you really need the full tomographic coverage to start clearly seeing structures inside the mouse for the optoacoustic imaging case. So of course, it's clear that you better get a scanner with the best tomographic coverage for your application. But this could quickly become quite expensive as you need a transducer array with lots of elements, and then also add, all the multi-channel electronics to acquire all this crazy amount of real-time ultrasound data. So if you want, if you can't afford it, then a possible solution could be to acquire data with less channels. For example, if you do some sort of undersampling of the channels. So of course, the resulting images in this case will have artifacts because you discarded a lot of important data points in the tomographic data set. But maybe you can still recover it, the image quality, by means of, if you're using deep learning. So the idea would be to train the algorithm on fully sampled data from this expensive scanner and then apply it to undersampled data. And here at the bottom, you see the results. So most of the artifacts from the undersampled data get removed, even for the case when you do 16 times undersampling. And you can also apply the same concept to the case of limited view imaging with the linear array transducers. So after training the algorithm on the full tomographic data, deep learning manages to compensate for the limited view artifacts to a large extent. And of course, the linear array images here still don't look the same as the gold standard data, original data, but still much, much better than just uncorrected images in this case. Okay, let's go back from the algorithms to applications. So another area where imaging speed of optoacoustic is very important is obviously cardiac imaging. So the mouse heart is beating 300 or 400 times per minute. So if you want to visualize the heartbeat in 3D in real time, your imaging modality must be quite fast. And here you see an example of 4-dimensional optoacoustic imaging of the mouse heart at 100 volumes per second. So this speed is sufficient to also visualize not only the heartbeat, but also perfusion of the indocyanin green contrast agent, which is injected into the blood and propagates through the heart chambers. And in this way, you can start characterizing some functional parameters of the heart, like the pulmonary transit time or some nonlinear parameters of the heartbeat. And this allows to clearly differentiate between abnormal heartbeat in, for example, infarcted heart versus healthy heart based on those functional parameters. So you may wonder that, this was already quite fast imaging performance, but it's not always sufficient. For example, what if you want to track a free behaving animal in 3D? So for this, you may need an imaging speed of maybe even 1,000 volumes per second, which would be quite problematic and demanding just in terms of the insane amount of data throughput that you need. So to solve this problem, we applied a compressed sensing approach where we shoot the laser pulse several thousand times per second. But for each pulse, only part of the transducer array elements are recording the data simultaneously. And for the next pulse, it's a different random subset of the elements acquiring the data and so forth. So for the image reconstruction, we developed a special type of a total variation-based reconstruction approach in a combined space-time domain. And here you see a movie of performance of this imaging approach. So basically, you can see images of a zebrafish swimming in a small aquarium rendered at a speed of 1,600 volumes per second. So here you see milliseconds running. So it's slowed down more than 10 times. And the same method is also important for some other cardiac applications. So, for example, what if you want to look at to sense mechanical wave propagation, mechanical wave propagation in a beating heart? So we demonstrated on a model of Langendorf model of an isolated heart, which is perfused and it can beat freely. So it doesn't have blood, which was important for our application because whole blood absorbs too much of a signal, so we could not see the whole heart in this case, so we isolated it. But the 1.6 kilohertz 3D frame rate allows you really to look at the different phases of the mechanical wave propagation through the heart. And here you see the phase map for the 7 hertz heartbeat component. And we can track the spatial propagation of the phase velocity vector. Now, if you applied it to an arrhythmic model of the heart, where, for example, you induce arrhythmia with some electrodes, so you can nicely visualize the phase singularities in the heartbeat that correspond to the so-called spiral wave reentry, which happens during the cardiac arrhythmia. So here you see this point where the wave, the mechanical wave, basically spirals around this point, and you really need very high imaging speed to visualize something like that, especially in the very fast beating heart of a mouse. So maybe a few words about multimodal optoacoustic imaging. Obviously, as I shown previously, you can combine optoacoustic imaging with ultrasound imaging because you already have an ultrasound array in your system. But you can also do it with optical imaging or fluorescence imaging. So, for example, if you already have an illumination source and then you detect the fluorescence responses from the tissue together with the optoacoustic responses, then you basically have a hybrid fluorescence optoacoustic imaging system. So for this, we developed those special kind of fiber scopes that can be inserted through the ultrasound detection array. And in this way, you get both images of blood contrast from a tumor, for example, here shown in red. So they are from optoacoustic imaging, and together, you can also get the fluorescence contrast by using the fiber scope. And maybe the most exciting development of the last 2 years is the really hybridization of optoacoustic tomography with magnetic resonance imaging. And we did it especially for functional neuroimaging applications. So we demonstrated it on imaging of brain responses to peripheral stimuli in a mouse. And to do that, we designed a special kind of ultrasound array, which is magnetically compatible and can go into the bore of 9.4 Tesla MRI. And they don't disturb each other. So the optoacoustic doesn't disturb the MRI, and it can acquire images simultaneously. So now we start looking into all the hemodynamic responses, both from the bold contrast of fMRI and also oxy and deoxy contrast of optoacoustics, and the results are quite exciting because we now can really correlate one-to-one this mysterious bold contrast to another modality that records simultaneously both oxygenated and deoxygenated components of the hemodynamic signal. Maybe last thing I want to mention is to talk about clinical optoacoustic imaging. So you're probably wondering whether, you know, all this basic research type of stuff that I previously showed can ever go into to the clinics. And the answer is definitely yes. So it's actually already happening. There are many ongoing clinical trials also in Europe, US and Asia, using handheld optoacoustic imaging technology to diagnose a variety of patients with different indications. So for example, breast cancer, atherosclerosis, inflammation, bowel diseases, muscular dystrophy, lymphedema, and so forth. I'm just going to show you a few interesting examples of promising clinical applications. So one comes from a clinical group in Erlangen in Germany, who is interested in early diagnosis and staging of Crohn's disease. So Crohn's disease is an inflammatory bowel disease, and to diagnose and stage the disease, they used a commercial version of one of our EMSOT scanners, which is combined with concurrent B-mode ultrasound imaging. And they also did transabdominal evaluation of intestinal inflammation in 100 patients with Crohn's disease. And then compare the non-invasive EMSOT results with clinical endoscopic and histological scorings. So in the study, they were able to show that active Crohn's disease stages could be distinguished from remission entirely non-invasively using a handheld EMSOT imaging scanner. And now there are phase 2 multicenter clinical trials going on in Germany and Italy to basically approve this approach as a diagnostic procedure to be reimbursed by the health insurance providers. So as I previously shown, our volumetric scanners clearly showed like entire volumes in real time. So we now look into another indication, which has to do with carotid artery disease. So it usually appears those plaques in the bifurcation area of the human carotid artery. And the basic idea is that optoacoustic imaging would be sensitive to hemoglobin, but also the lipid content of the plaques, which potentially could be a good biomarker of the plaque vulnerability and also risk of rupture and stroke. And these published results that you see here are from healthy volunteers, but we are also working together with the neurology department here at the university hospital to image atherosclerotic patients. And the results are quite promising. And maybe last example I wanted to show you, it comes from another group in Germany, in Essen. And they're interested in non-invasive detection of metastasis in sentinel lymph nodes. So those metastases appear as a result of spread of melanoma, the skin cancer. And it's very difficult to detect it non-invasively because those melanin deposits are very small. So you really need to go in and do invasive biopsy and then slice the lymph node. And sometimes you miss it, sometimes you overdiagnose, sometimes you are not able to do it. So the idea would be to do it non-invasively, and optoacoustic is actually good modality to try because it's melanin absorbs a lot of light. It's dark, so it detects a lot of signal. So in this study, they showed that EMSOT could identify cancer-free sentinel lymph nodes in vivo without a single false negative. So basically, they scanned 189 lymph nodes. So without single false negative means 100% sensitivity. The specificity was a bit lower, but this really allowed to confidently rule out the presence of metastasis in melanoma patients. So now you can rely on it not to do biopsy, invasive biopsy, and you would still be accurate in this kind of diagnosis. Okay, so maybe time is up for me to summarize. So what I've shown you today is that optoacoustic imaging is a multi-scale method that can take you all the way from high-resolution microscopy to centimeters of penetration in human subjects using tomographic approaches. It's safe for human use because it uses non-ionizing radiation in the visible, and well, mainly in the near-infrared spectrum. The imaging speed could be very fast, so even 1,000 volumes per second is possible over limited fields of view. You can do label-free imaging of intrinsic tissue contrast that has to do with angiogenesis, for example, hemodynamic changes, oxygenation, some other useful content of melanin or lipids. It's possible to use molecular contrast agents, at least in mice. For clinics, it's a bit difficult because you need FDA-approved agents. The sensitivity in this case would be fairly high. It's still not comparable to PET, positron emission tomography, that goes up to like femtomole, but we can work with, for example, picomoles of agents still in vivo. And I've also shown you a broad palette of preclinical and clinical applications in cardiovascular disease, oncology, brain research, and others. There are many still a lot of challenges to be addressed. So the technical usability is still not comparable to conventional clinical modalities like ultrasound or MRI. Penetration depth is a big issue, so we are able to go through a few centimeters, but in human applications, that's quite limiting. Image quantification is still an issue, so there's a lot of algorithms, algorithmic efforts invested into this. Contrast agent selection is pretty broad because you can use optical agents, but ideally you would use some dedicated agents. The clinical evidence is still also quite sparse, so not many clinical studies available. And additional big issue is that you eventually need to go and standardize the approach, which is still not happening, at least not at the right pace, I would say. Okay, that was it. I would like to obviously thank the brilliant and motivated students and postdocs in my lab who did all this great research, and also our collaborators and the generous grant support. And I'm ready to take any of your questions. Thank you.
Thank you so much, Tanya, for the wonderful talk. Those images are amazing. Now it's open to questions.
I can ask one.
Please.
Thank you for a very delightful talk, Dr. Razansky. So I have one question that, so you talked about optoacoustic mesoscopy. So I was wondering, like, what's the difference between optoacoustic mesoscopy and the acoustic resolution photoacoustic macroscopy?
It's the same thing. I just call it this way, not to confuse the two, but it's indeed called this way in some papers. But we do it for clarity purposes, to separate really mesoscopy from microscopy. So microscopy only refers to the optical resolution methods.
Okay, yeah, thanks.
May I have a question, please?
Sure.
Daniel, good to see you. Congratulations. Your results are really brilliant, and your lecture demonstrates that optoacoustic has great prospect, especially in research. But my question about clinical application. I'm interested in tomography. you told that in real time tomography required many detectors. And the question, what about miniaturization of the device? Is it possible to to create this detector in a realistic device? What the dimensions?
Mm. Yeah, excellent question. There are many approaches how to miniaturize it. One way is to piggyback on the existing ultrasound transducer technology that is evolving very fast. So there are many new technologies where you can condense a large number of elements, for example, using CMOT transducers. Another way of doing it is to switch to optical detection of ultrasound. So if you use some optical interferometry approaches, so the effective size of each detector could be squeezed to a 100 micron range or something like that. The issue is then how to multiplex it and parallelize it still. But in terms of making it miniature, there are different ways of doing it. Yeah.
Thank you.
Okay, any other questions? So I can ask, so you mentioned this single sweep system. You didn't spend too much time on that. Can you just go into some details how you can finish it with single sweep?
Yeah, so the trick was to move this large spherical array mechanically along the mouse very fast. So basically, it goes with a speed of 80 millimeters per second, and then you can do it in a matter of 1 or 2 seconds, the whole mouse. But at the same time, if you don't rotate it around the mouse, just move it linearly, then you don't have the full tomography of like having all the tomographic view. And the main problem is not just the lack of ultrasound detectors from all the angles, but also illumination, because you really need to illuminate the entire mouse to get signals. So we basically designed a special illumination system that goes around the mouse from all projections. And then we could cover the entire mouse body. Of course, the image quality is not still the same as if you go also like rotate around the mouse, but it's reasonable. So it's a good compromise between, you know, the field of view and the speed.
Right.
Yeah.
So you mentioned the MRI optoacousticimaging integrated system. So what kind of transducer are you using within the MRI? I assume you have some special non-metal transducer, right?
Well, in terms of just the geometry of the transducer is quite similar to all the other spherical arrays. So it's a spherical array.
How about the materials? Because if you have any, like these piezo transducers, sometimes mostly has metal on the surface, and can you use that in a MRI simultaneously, or?
Well, the piezoelectric material itself is not magnetic,
Right.
but you should not have any metallic coatings on the transducer. So it won't be a good idea to have gold or, you know, this kind of stuff. And also we got rid of metallic casing, because I mean, you would usually have a transducer with this kind of casing to reduce the interference, like from the environment.
Right.
So you need to use some other shielding materials like copper, you know, to get rid of electromagnetic interference without using stainless steel or some other magnetic. Yeah.
I see.
But other than that, there were no big like interference, at least not for the MRI. The optoacoustic detection suffered from some MRI sequences still. So in case you have some fast gradients, it induces some currents, you know, on the wiring, and you get huge artifacts. So we had to deal with that with some algorithms and also some hardware modifications. So it was quite some work. Yeah.
I see. Thank you. Okay, any other questions before we close the seminar?
If possible, one more question, Daniel. When
Sure.
you are talking about atherosclerosis application, what do you mean? The plaques in the artery or something else?
Yes, exactly, the plugs, but not just the plugs. So the main risk is not from the plugs, which are usually calcified well and they're not at the risk of rupture. They, you know, the main problem is to decide when to intervene, because when the plug becomes vulnerable, like this kind of jelly
Yes,
type of plug. So that's the main
correct.
risk.
Yes, it's one of the problem, but calcification is also very important. Can optoacoustic recognize and separate hydroxyapatite part of the plaque, fibrous? So can optoacoustic recognize the distribution of plaque?
Right. Well, so the things you mentioned were not demonstrated, so I cannot tell if it's possible or not. But generally, our hope is to detect lipids, because lipid content is usually considered as a risk factor.
Yes.
And it's not easy to detect it with with ultrasound. It's not having the right contrast, but even with the modern MRI. It's very ambiguous to detect lipids.
Correct.
The second, and you know, optoacoustic can sense the molecules of lipid because it has a specific spectral signature. The second biomarker could be the blood itself, because plaques often have, you know, all this angiogenesis going on, and so maybe this could be another indicator. Yeah.
Thank you.
Okay. Thank you all for attending the seminar. Thank Daniel for the wonderful talk again. For those who want to meet with him, plan to meet with him, we can stay on the same link. And thank you all for attending the seminar.
Thank you so much.
Thank you very much.